Hey guys, Mr. Hyatt here. This is part 3 of the Chapter 43 AP Biology lecture on the immune system. We're picking up here talking about B cells. Humoral response is that's the left side of the diagram we just talked through. That's going to be characterized by a secretion of antibodies by the B cells. B cell activation is going to be aided by cytokines and then binding to the helper T cells. Remember, helper T cells help everybody. And then clonal selection of the B cells generates the antibody-secreting plasma cells, which are the effector cells of the humoral side of things. Here's a picture that shows that. We've got our class 2 molecule here. Let me fix my text box again. Sorry about that. Okay, so we've got our antigen-presenting cell. There's our bacterium getting into the cell. Here's our activated helper T cell. We've got our B cell that's now binding, that's creating these antibodies. We're making clones of those plasma cells. That's that clonal selection. And then they are going to lyse and they're going to release these antibody molecules that are then going to break down different bacteria that are presenting themselves. We're also going to have memory B cells that are created to accelerate that process next time. So this just sort of walks through that. I already covered it. There are 5 major classes of antibodies, and they're different based on their shape and their function, of course. Excuse me. Polyclonal antibodies are going to be the products of many different clones of B cells following exposure to a microbial antigen. That should make sense because it's polyclonal. Monoclonal antibodies are prepared from a single clone and grown in culture. Those are going to be we grow those in the lab, if that makes sense. I know you can't read this. It's in your book. Make sure that you can summarize each step in this table. So there's our IgM. You don't need to be able to, I misspoke. You don't need to be able to fill in every piece of this table. As we get closer to test day, I will tell you specifically which of these that you'll need to commit to memory. For now, you need to be able to recognize them. So if I put this shape up, you should be able to recognize that as an IgE. You should be able to recognize that as an IgA, IgD. So what role do antibodies play? Well, you can neutralize a pathogen, which is essentially connecting it to an antibody, and then you can opsonize it, and that's where it's sort of like an enzyme idea applied to your immune system. So when antibodies bind to antigens, increase phagocytosis, that's called opsonization. The antibodies are going to stick together into complexes, and they're going to, here's what I'm talking about, okay? So we've got a virus that's been neutralized. It's just, it can't attack the cells. In terms of opsonization, now that we've got these antibodies bound to this cell, macrophages recognize, okay, that's been neutralized, I need to destroy it. It's been marked for destruction. And then we could activate a system that's going to attack, kind of like the idea of multiple opponents. You're attacking as a group rather than just attacking as an individual. Strength in numbers. So active immunity develops naturally in response to an infection. So it can also develop in terms can also develop following a vaccination or an immunization. An immunization, a non-pathogenic form of a microbe or part of a microbe, elicits an immune response to an immunological memory. So we're basically going through the first exposure on our own. We're jump-starting it. Now, notice it's a non-pathogenic form of the microbe. Could be that it was a pathogenic form that we've heat-shocked. We've boiled it. We've killed it. It's still functional. I mean, it still appears functional, but it's not. It still has the same receptors and all those sorts of things, but it's not alive. Could be a modified, maybe a genetically modified pathogenic form that's now non-pathogenic that, again, presents the same receptors but lacks some of the DNA that it requires to infect us. There are a couple different ways that we can do those immunizations, and different. We do both. We do multiple types of immunizations depending on the bacterium or the antigen, the pathogen, something along those lines. So passive immunity is going to provide immediate short-term protection, conferred naturally when IgG crosses the placenta from mother to fetus, or when IgA passes from mother to infant in breast milk. So can be conferred artificially. You can use an antibody injection into a non-immune person. You can transfer immunity that way, like almost like an antibiotic. But again, it is conferred naturally. Passive immunity comes from mom in one of those two ways that I just mentioned. When we see immune rejection, anytime we see transplants, we can essentially, our immune system recognizes these cells as non-self, and they're going to start destroying them. So many times we have to take immunosuppressants so that our immune system doesn't work well, or we might have to go through radiation to try to damage some of those cells. But just another sort of iteration and another sort of example of what these things can do when they work well. Another application of this chunk of material is our blood groups. So the antigens on red blood cells determine whether a person has type A, type B, type AB, or type O. Notice that if we have type A, we have A antigen. If we have B, we have B antigen. If we have AB, we have A and B antigens. And O is the omission of all antigens. So there are 0 antigens. Antibodies to non-self blood types exist in the body. So if I have A antigens, so I have type A blood, then I'm going to have type B antibodies. So if I get type B blood injected into me, transfused into me, then my body's going to recognize that as not supposed to be there, and it's going to get rid of it. Before we move on to tissues and organ transplants, think about what that means for host and donor compatibility. If you have neither antigen, you can probably donate to anybody because no one is going to recognize that blood as non-self. If you have both A and B antigens, you probably don't have any antibodies, so you can probably accept anything. MHC molecules are different among genetically non-identical individuals, so identical twins are going to have identical MHC complexes, MHC molecules. I don't know why I said complexes. But the differences in those molecules are going to essentially stimulate the rejection of organ transplants and tissue grafts. That's why you look for someone who's a really close relative when you're looking for a donor. Again, that's going to ensure that your MHC tissue types are really well matched. So we've kind of already talked about those second two bullet points. Don't need to rehash them again. So disruption in the immune system function can cause or make worse a disease. Some pathogens have essentially evolved to make our immune responses weaker. So what consequence will this have, and what's the adaptive value to the pathogen? Well, the consequence is that weaker things, so think about something like HIV that suppresses our immune system. Most people that die with HIV die from some relatively minor disorder, something just some sort of infection that someone who didn't have HIV would be able to fight off. Adaptive value of this ability of the pathogen is it doesn't kill the host, but it weakens their immune system and allows them to divide, to multiply, to make copies of themselves and spread themselves around using the host's metabolism and the host's stuff, basically. So think about these three. This is something I'm asking you to look at with your debrief. So there's a delicate balance in your immune system. If those are thrown off, then you're gonna see changes. Examples are allergies and autoimmune problems. I've got examples of both of those. So let's talk here, and then I think I'll be able to bring it back around to exaggerated, self-directed, and diminished immune responses. So allergies are exaggerated responses to antigens that we call allergens. In localized allergies like hay fever, IgE antibodies produced after the first exposure to an allergen attached to receptors on mast cells. The mast cells release those histamines, causing many of the symptoms of allergies, runny nose, sneezing, all that kind of stuff. Remember, histamines dilate blood vessels. So they're going to cause inflammatory responses all over the place when we have these histamines. So why do so many allergy medicines contain antihistamines? Because they need to shut down the histamines. That's the problem. So here's the mast cell that's got these IgEs. They're connecting with the allergens. The allergens are binding to these ports, these receptors, these presenting sites, and we're creating histamine and we're releasing it, and that's causing blood vessels to dilate and all those types of things. The next time the allergen enters the body, it's going to bind to those IgE cells. We're going to release other mediators and histamine. It's going to cause the vascular symptoms that we see. So an acute allergic response, so a local allergic response, can lead to anaphylactic shock, which is a life-threatening reaction that can occur within seconds of allergen exposure if you have a really strong allergy to something, like I do with poultry. I can go into anaphylactic shock really easily if I inject or if I eat turkey, say for example for Thanksgiving. So quickly, let's talk autoimmune disorders. Immune system is going to lose tolerance for self, so it's not going to recognize your own cells. Essentially, your body is attacking itself. We've already talked about one of the types of diabetes that is autoimmune. Systemic lupus is autoimmune. The research shows that once you have one autoimmune disorder, you tend to have other autoimmune. You're more likely to develop other autoimmune disorders. HIV is an example of an autoimmune disorder. Again, it's just your body attacking itself. Moderate exercise does improve immune function, and psychological stress has been shown to disrupt hormonal, nervous, and immune systems. So all three of the body systems that we struggle in AP Bio, stress jacks with them in a number of ways. So if you want to be healthier, exercise a little bit more and de-stress. A lot of times those two things go hand in hand. Immunodeficiency diseases are going to be things where your immune system is deficient. Acquired immunodeficiency syndrome is called AIDS. We know lots about that one. But anytime you've got an immunodeficiency, your immune system is just not quite working right. Pathogens have evolved mechanisms to attack multiple immune responses. HIV is really good at doing what we're seeing here, where we're varying our antigens, and it's also really good at mutating and creating new antigens. Flu virus mutates rapidly. That's why we have to have a new flu virus a new flu shot every year. It doesn't carry through because the receptors are different. The antigens are going to present differently. Swine flu, bird flu. We can exchange DNA with animals. It's really common in domesticated animals. And then we're unable to recognize the new viral strain because it looks just like the old viral strain. Here's kind of what I'm talking about, the sleeping sickness. We've got 3 variants, and each variant, the exposure causes a very first exposure kind of a curve. There's a lag, and then we build antibodies, and then we are exposed, and there's a lag, and we build antibodies. They're changing every time. We're not seeing that same graph where we see less lag and a higher ceiling. We're seeing the same graph over and over. That's part 3. I will wrap the section up, wrap up the lecture in part 4 of the chapter 43 lecture.